We're talking about proteins. Proteins are a type of macronutrient, just like carbohydrates, just like fats. And proteins come in two major forms. We have structural proteins and we have functional proteins. So the structural proteins, their main job is like to build the different shapes and forms of the body, hold things together like connective tissue and bones and hair and fingernails, that kind of thing. Functional proteins, they are, like their name says, they're performing some type of function. So these are proteins like protein hormones, antibodies, gates or pumps that we have in our cell membrane. These are all types of functional proteins. Now, one of the things that you're aware of is that proteins are built from separate amino acids. And these amino acids are all going to contain nitrogen, and then many of the amino acids also contain sulfur groups. So the sulfur and the nitrogen, our main source of those in the body, come from the various different amino acids. So we have 20 different types of amino acids that we find in each one of the proteins of the human body. So a protein is usually about 1,500 amino acids long or larger. Now if it's less than 1,500 amino acids, let's say maybe it's 100 amino acids, instead of referring to it as a protein, which can also be called a polypeptide, this smaller group of amino acids would just then be referred to as a peptide. So it's almost like it's a fragment of or a portion of a protein, okay? Now, each amino acid, like you can see in the picture here, they each have a carboxylic acid group. So if you remember, carboxylic acids are going to be that C double bond O, OH group. This is our carboxylic acid. And then every amino acid is going to have an amine group. Okay? But to make each of the 20 amino acids a bit different from each other, then we're also going to have what's referred to as an R group, or this is also called a functional group. So all of our amino acids have the amine group, they have the acid group, and then they have the R group or the functional group. And the functional group is different between every single one of the 20 amino acids. And in order to have a functioning protein in the human body, somewhere in, let's say, that 10,000 amino acid long chain, you have to find all 20. If you don't have all 20 of those amino acids, then the protein will not be able to take on the right shape and therefore won't be able to have the right function. Now also remember that when we take amino acids and bond them together, we take one amino acid and take its amine group and bond it to the acid group of the other amino acid. And when we bond them together, we form a bond called a peptide bond. Okay? So peptides are typically somewhere around 50 amino acids long, maybe smaller. If you have something that's just called a dipeptide, that means all you did is put two amino acids together. If you have a tripeptide, now you've bonded 3 amino acids together. If you have a polypeptide, this is typically hundreds and thousands of amino acids long, which is our protein. So our proteins are typically referred to as polypeptides. Now, to put 2 amino acids together, when you form that peptide bond, one of, you might call it like the waste products, or one of the other products that form is water. So a lot of times we can say this is a condensation reaction because when water is formed in that reaction, it's given the word condensation. Now, when you split the peptide bond, you actually use some water to be able to split this, and so we call this a hydrolysis reaction. So if water is involved in the splitting of the peptide bond, you have a hydrolysis reaction. Okay, so now we have these 20 amino acids, and we can break them down into basically two groups. We have one group of amino acids that are called the essential amino acids, and then we have another group which are called non-essential amino acids. And 9 amino acids fall into the essential category, and 11 fall into the non-essential category. So somebody tell me, what does this term mean? What does de novo synthesis mean?
To make something out of nothing?
Yeah, to turn something into something else. Okay? So, and really it means to make something out of nothing. But we, in the body, we take something and we turn it into something totally different. So the non-essential amino acids, we call them non-essential because we can do this de novo synthesis where we take something that's in the body, some product in the body, and we can turn it into any of these 11 amino acids. So that we don't actually have to eat them in our diet. But those 9 those 9 essential amino acids, it then becomes essential for us to eat them in our diet because we can't make them through de novo synthesis. So if you have a person who's a carnivore and they eat meat, anytime you have somebody who's eating meat, doesn't matter, fish, chicken, cow, whatever, as long as they're eating meat, that meat has all 20 amino acids in it. So their diet is going to provide them with all the amino acids that they need to be able to build all the proteins that we need in the body. But let's talk about somebody who might be vegetarian or even someone who is vegan and they're not eating any type of animal products at all. No cheese, no milks, nothing, no eggs. And so one of the big issues for them is that not all plants provide all 20 amino acids. So when you're talking to someone who has that eating lifestyle, what you have to make sure is that they are well aware of their nutritional needs as far as protein is concerned. They need to study it, they probably need to get some books, maybe even talk to a dietitian or a nutritionist and find out what combinations of food that they need to eat on a daily basis. Because our body is constantly tearing down proteins, and we're going to talk about this a little bit more, but we're constantly tearing down protein, which means on a daily basis we constantly have to build protein. And so if I don't have enough of these amino acids in my diet every day, then I have a serious problem. So I tell the story typically in physiology, so for some of you who didn't have the class, I just want to tell it again. In the 1960s, there was a manufacturing company that came out with this diet formula, and it was kind of like protein powder like we have today. And the whole idea was you took this powder and you put it into water and you stirred and then you drank this 3 times a day and you didn't eat anything else. And it will help you lose weight. And it's like, well, no kidding. That's all you do is drink this 3 times a day. People are going to lose weight. Except people die. Because look at this list here. These are our essential amino acids that we have to eat. And one of these amino acids is tryptophan. And when this company was making this powder, they neglected to put that amino acid into the powder. Which means the people who were drinking this stuff and not eating anything else, not eating any kind of meat products, nothing, they didn't get that amino acid in their diet. And the lack of that one amino acid would not allow them to build any proteins in their body. So then if you think about the fact that the heart is beating all day long and that some of that protein falls apart all day long, if you don't have all 20 amino acids in your diet, you cannot replenish those proteins in the heart. And so what they saw was a lot of people having heart attacks all over the United States. So if you see any of these kind of powdery, lose weight, slim fast types of things nowadays, they'll tell you, drink this for breakfast, drink this for lunch, and then have a healthy dinner. And there's usually a big piece of chicken or something in the meal, the picture, so that you should be eating some meat so that you can get all 20 of your amino acids, because then this becomes a big problem. Now there's another group of amino acids that sometimes they'll call conditionally essential. This is when our body has something wrong with it. We become ill, or there's a genetic defect and I don't have the right enzymes, and now I can't do certain de novo synthesis. So I might not be able to make certain proteins in my body, therefore I need to make sure that I eat them. Okay? And so then that becomes a non-essential amino acid that became what they call conditionally essential. Yeah?
So you said all meat has all 20?
All 20. Yep. Because think about it, our muscles are made up of protein, so they'll have all 20 amino acids in it, and that's what we're eating when we eat meat. We're eating the muscle of that animal. All right, so let's just go through this real quick. You know that when amino acids join together, you get those peptide bonds going on. There are shapes that our proteins are going to form into. So we have what's referred to as the primary shape or the primary structure of a protein. And this is where you're just taking amino acids and bonding them together, and you're getting this long chain of amino acids. Almost looks like you have this big strand of pearls type of thing. Okay? This is your primary structure. The secondary structure is going to occur because when we look at the amino acids, they have some side chain molecules. And some of these are hydrogens, some are nitrogens, some are oxygens, and they like to bond to each other. So you form this long strand, and some of our proteins will just stay as long strands. That's it. That's their job. They don't continue to change shape. But because of these side oxygens, hydrogens, nitrogens, they begin to bond to each other in this strand. And it's almost like you take that string of pearls and you start winding it around and around and around, and you get this long alpha helix structure. And this is our secondary structure. Now, if the protein takes on a primary shape or a secondary alpha helix shape, these proteins are all going to be used by the body as structural proteins. So remember, those are like the connective tissue, collagen, the proteins in bones, the proteins of our hair, that's typically a secondary structure, your fingernails, those types of things, to give us the form and shape of our body. Now, some of those side chains will continue to bond, and by the way, most of this bonding with the side chains are hydrogen bonds. And as those side chains continue to form these hydrogen bonds, the alpha helix starts to fold in on itself. And you get your tertiary structure. And what do we call a tertiary structure? A glob. Or a globular protein. If I take 4 globs and bind them together, I have a quaternary structure. And what's the only molecule in the body that is 4 globs?
Hemoglobin.
Hemoglobin. Excellent. So then that means that our tertiary and our quaternary structures, these are going to be our functional proteins. Which then also means your protein hormones, your antibodies, those gates that are found in the cell membrane along with the pumps, all those things are all tertiary. Enzymes, those are all going to be tertiary proteins. Because the only functional protein that's quaternary is hemoglobin. Everything else is tertiary. It's the shape of the protein that allows it to do its job. And we know that the shape occurs because hydrogen bonding is occurring. What happens to a protein if I just break a couple, not very many, but a few hydrogen bonds? What will happen to its shape? It changes. Yes, it changes. What happens to a protein if I break a mess of hydrogen bonds? It denatures. So to denature a protein means that I break so many hydrogen bonds in that protein that it not only loses its shape, but it also loses its function. So breaking a couple hydrogen bonds causes the protein to change shape ever so slightly. Breaking a lot of hydrogen bonds causes the protein to change dramatically, and then that protein also can no longer function. It can't do its job anymore, and we say that the protein has denatured. Now, one of the ways to denature a protein is heat it up. Like if you take an egg, which is made out of tons of protein, and you cook that egg, it doesn't look the same, does it? Totally different. And the reason it takes on a different shape is because you denatured its proteins. In the human body, if we start to get a fever and we start to heat up, we can also see that lots of hydrogen bonds in our proteins break and our proteins start to denature, which is why having a fever is so dangerous. And if you remember, we got to get our temperature up to about 40 degrees Celsius, which is the same thing as 104 degrees Fahrenheit, in order to start denaturing those proteins.
You said children's was higher, like 106?
Yes. So kids, we don't really understand, typically around the age of 11 or below, they have some kind of protective mechanism that actually allows them to get a little bit higher fever before we see their proteins denature. So they typically get to about 106 before their proteins denature. This is one of the reasons right here why they developed vaccines. Because if you look at the measles, the mumps, and rubella, they were major killers of children. Because when you get these diseases, it is quite common for the temperature of that person to go up to 108, 110. And now we see people deciding not to vaccinate their children. And for you guys, when you start working in the hospital, you're going to see these diseases again, which have not been seen very often in the United States at all, because vaccination was just what you did, okay? And people are deciding not to do that. I'll tell you from personal experience, I would rather have been vaccinated and possibly had autism than have to go through the measles, mumps, or rubella, because it was horrible. And by the way, I had the measles 5 times. So just because you get it once does not mean your immune system actually gets the message, and you can get it over and over again. My brother had the mumps at the same time that I did. He was much worse than me. His fever actually went up to 112. They thought for sure that he would be mentally retarded when they're done. He is my brother, so he probably is. But he had all kinds of other defects as well. When his permanent teeth came in, they all had pitting in them, because they're already in your jaw, and so it denatured all of his permanent teeth. He had other issues because of the proteins that were denatured throughout his body. For whatever reason, I'm not sure why, he was able to have children. And usually for the male, when they get the mumps, one of the first things that happens when you get that high of a fever is that it creates problems in the testicles, and so they can't produce sperm. He's got 5 kids, so that wasn't the problem for him. So
why did you get him 5?
Say it again?
How did you get it 5 for him?
My immune system didn't make memory cells. So your immune system is supposed to make memory cells and go, oh, I recognize that as a disease, let me kill it off really fast. Because that's what a vaccine is. You get a little bit of that virus, and you get a little sick, and your immune system defeats it because you only have a little tiny bit of it, and then you create memory cells. And so as soon as somebody sneezes or coughs on you and that virus gets in, your memory cells are right there to mount a defense to kill off that virus. Well, apparently my memory cells were a little on the lazy side, and they just didn't do their job, and so, woohoo, I got to have it 5 times. But that does happen. That does happen to people. So people will have, like for instance, they'll have chickenpox more than once. That's not unusual, because their immune system just didn't mount a good enough defense. All right, so something else that denatures proteins is change in the pH. Now, typically for the human body, if our pH drops, it goes too low, this will cause our proteins to denature. We rarely ever see the human body with a pH that's too high. Now, let me just ask you, a pH that's too low, is that an acid or a base? Acid. Excellent. So we become, when we're sick, we typically become more acidic. So that's what usually is going to happen in the body. So we typically see proteins denature at an acidic pH, below 7.35. We rarely become basic, but if we do become basic, if we get to 7.6, okay, because we can go up to 7.45, if we get to about 7.6, we'll start to see proteins denature as well. But it's pretty rare. Another thing that can make proteins denature is agitation, and that doesn't mean you get mad at your teacher. That means literally you have to shake them back and forth, okay, or like you see with the wine that they're going around and around, that actually does break down proteins. And in wine, it breaks down proteins and releases the odor, which is quite interesting. Okay? Now, why is it, to kind of go back a little bit, why is it we would want any proteins to break even the slightest, tiniest amount of hydrogen bonds and do a little shape change? Why is that beneficial? What is happening to the protein when it has just a slight shape change? It changes it to something else, and it has a different function. So for instance, we know that enzymes are proteins, and enzymes do what again?
Lower the activation.
They catalyze a reaction by lowering the activation energy. Perfect. And basically what that means is the enzymes take 2 substrates and smack them together. Okay? But they can only do that because they get a shape change. So we break a few hydrogen bonds, and then the enzyme changes shape to bring these chemicals together. What do we normally have to add to an enzyme to get it to change shape?
Energy.
A phosphate. And where do we get that phosphate from typically? ATP. Very good. You didn't know you were going to have to do a physiology quiz today, huh? All right, so now, When we're going to digest enzymes, or excuse me, I should say proteins, in the body, digestion is going to start in the stomach. So there's really nothing that helps us to digest the protein per se in our mouth. There's no special enzymes or anything like that. We're just kind of chewing it up, and then it gets into the stomach. And the big one in the stomach is hydrochloric acid. That chemical can pretty much start dissolving anything. But we also have a chemical called pepsinogen. So I'm going to go to this. Do you remember a picture kind of like this with those good old-fashioned gastric pits? All right, so let's talk about these just for a second, because this is very important in protein digestion. So you have this gastric pit. And it has all these cells around it. And each of the different types of cells are at their own level, and they're always at the same level. So the guys down here are typically called chief cells. And chief cells secrete what? Do you remember? They don't secrete hydrochloric acid. Those are the next guys, which are the parietal cells. They're going to be responsible for the hydrochloric acid. So what are the chief cells making? Charlia, what are they making?
Pepsinogen.
Perfect, that's exactly right. So these guys are secreting something called pepsinogen. What does the OGEN stand for?
It's inactive.
It tells us that it's not active yet. Because if we had an active enzyme inside the cell, it would dissolve all the proteins in the cell and destroy the cell. So we want it to be inactive until it's secreted into the gastric pit. Now the parietal cells are secreting hydrogen ions, and they're secreting chloride ions separately. So that what we see is you have these hydrogen ions plus chloride ions that mix together to form our hydrochloric acid. And this is what gives our stomach that pH of about 1.5 to 2, something like that, which is pretty acidic and can digest just about anything. And it's going to mix with pepsinogen. Okay? Our inactive enzyme. And when hydrochloric acid mixes with this, we're going to activate this enzyme. And now we call this pepsinase. Now your book probably calls it pepsin, but they have recently changed the name, which was just smart. We want the ASE at the end. The job of pepsinase it's going to take proteins that come into our stomach from our food and break them down into their original individual amino acids, which are very easy to absorb through the wall of the stomach, but especially they're absorbed through the small intestine wall. So this is the major digestive process for our proteins. Lots and lots of it is happening inside the stomach. Now there's also a little digestion in the small intestine too. So in the small intestine, remember, we're secreting that hormone from the wall of the small intestine, which is called cholecystokinin. Which is CCK. One of the jobs of cholecystokinin is to stimulate the pancreas. And the pancreas then also releases several different types of enzymes called proteases. So each of these enzymes has their own special name. I just want you to know that a group of enzymes that digest proteins, we call them a protease enzyme, okay? I don't want you to know every single name. There's a whole bunch of them released from the pancreas to help to digest all these proteins. So cholecystokinin says to the pancreas, hey look, release some of these enzymes. They're released. And now we're going to break down these proteins even more to make sure if they didn't get broken down in the stomach, they're going to get broken down into the individual amino acids so that we can absorb them into the bloodstream. Any questions so far? Okay, so I know a lot of that was just review, but hopefully it helped to kind of refresh your memory a bit. Now the amino acids, when they're absorbed through the wall of the small intestine, the first place they're going to go is to the liver. And the liver is going to make some decisions about these amino acids. So for instance, one of the decisions could be, do we need to convert these amino acids into glucose? Does the body need some more glucose? And amino acids can be converted into glucose in the body. Or the liver is going to make a decision and say, we've been eating a lot of protein over the last couple of days, and that bloodstream is just packed with amino acids. I don't think we need any more amino acids floating around in the bloodstream, and I'll tell you why you don't want too many in a minute. So what the liver will say is, okay, here's what we're going to do. We'll convert those amino acids into fat and store that puppy for later. Of course, one of the other problems is that sometimes these amino acids don't even make it to the liver. Instead, what will happen is they are then bypassing the liver and going straight into the bloodstream because we have what's called leaky gut syndrome. And we talked about that a little bit before. And of course now you have far too many amino acids floating around in the bloodstream, and they may not be broken down into individual amino acids, which then causes our immune system to go a little haywire and consider this as an allergen, and now we're going to try to attack just something that would be considered food. And of course that's not good for the person. Okay, so if an individual is eating sufficient carbs, amino acids, or excuse me, it should say insufficient carbs, amino acids can be converted to glucose, and the process is gluconeogenesis. That's the same process where we convert fat into glucose as well. So if I need to convert amino acids into glucose, or I need to convert fat into glucose, I use the process of gluconeogenesis. Interestingly enough, if somebody is super duper stressed out, you will see very high rates of gluconeogenesis. So let's say maybe you have a person who they're getting married at the same time they're buying a house, at the same time they're having a baby, at the same time other crap is happening in their life, and so there's a lot of stress going on there. You see a lot of gluconeogenesis, which also means that they lose a lot of weight because they're breaking down proteins, they're breaking down fats to use for glucose to get their body powered to get through this stress, and they typically lose a lot of weight during this process. But this is only with very high levels of stress. In order to have a lot of gluconeogenesis going on, you have to have lots of cortisol, lots of epinephrine floating through the system all at one time. Usually when we're only kind of under medium levels of stress, like sort of being a college student, all we want to do is eat all the time. And then instead of getting skinny, we just sort of keep widening and widening, which just really sucks. And we'll talk about why that happens too as we talk about proteins today, because they play a big part in that. Now, we have something called an amino acid pool. And this pool is really amino acids that are floating around in our bloodstream. So we talked about the fact that we're always having wear and tear on the amino acids of our body. So maybe you have a patient who's been burnt, or maybe they have a large wound that needs to be healed, okay? And there's a lot of amino acids that have been destroyed because of this. Because we have these amino acids that are floating around in our bloodstream, we use this pool in order to be able to repair. We really do not want to steal from other parts of the body all those amino acids to make repair. Because where am I going to steal from? I don't have anywhere like fat. I don't have anywhere to store amino acids. I store them as actual protein structures of my body. So if I have a large burn and I have to repair this with amino acids, maybe the burn has gone down to the muscle, I don't want to steal from other muscle to repair this muscle. So I have to eat a certain amount of amino acids to keep a certain amount of amino acids pooled in my bloodstream for daily repair. And usually we're talking about maybe 200 grams of amino acids a day. And what that means when you're looking at protein, it's like maybe eating twice a day enough protein that is about the size of a, bar of soap. Not as thick, but about the same size. That's really all the protein that we need in a day. Which is really weird because most people either eat much more than that or much less than that. And we really don't need a ton of protein. Now, our body doesn't care where it gets the protein from. You can get it from animal product, you can get it from plant product. It doesn't care as long as you've got those 20 amino acids. It doesn't care if you get it from white meat or red meat or pink meat or whatever it is. All our body cares about is, do we have all 20 of those amino acids in order to build this protein. Also, 50% of the protein turnover, that breaking down a protein, synthesizing the protein, this is all happening in our intestine, happening in our liver. And so when we break proteins down, and we'll talk about this, we break them down in the liver. And when we start building proteins up, we typically build them up in the liver. So the liver is very important in this protein production, which is also why when people have cirrhosis of the liver, they also typically have severe edema. And we'll talk about what the edema is all about as far as proteins are concerned. So you remember those processes of protein synthesis? We've got transcription, and we have translation, and part of translation is elongation, okay? So what's transcription? Say it again?
I said transcribing.
Transcribing, oh good, I like that. Okay, what's transcription besides transcribing?
Taking DNA to make RNA.
Okay, so I'm taking a gene, a section of DNA, right? And what does a gene code for?
Protein.
A protein. It's the code for a protein. All those bases are a certain code. So I'm taking that section of DNA and I'm making a copy of it, and we call it RNA. Okay? Specifically, what type of RNA?
Messenger.
Messenger RNA. That messenger RNA leaves the nucleus and goes into the cytoplasm. It hooks up with what organelle?
The
Rough endoplasmic reticulum and the ribosome. Exactly. So the ribosome is actually also a type of RNA, right? It was made originally in the nucleus. And then we have one more type of RNA. What do we call it? Transfer, or tRNA. And what is its job? transfers what? No. What's it transferring? Amino acids. And it's bringing those amino acids to the ribosome, and we're building this long chain of proteins, right? So what if I have this, and we'll go through this and then we'll take a little break. So I'm going to have this one section of DNA, and we'll say this is part of a gene, okay? So I have A, U, G, and then we'll say C, 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 G, C, A, 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 okay? So this is just our one section of a gene. Oh, I'm sorry, that can't be that. A lie, it's not. C, A, T, okay? And then I'm going to make a copy of this for messenger RNA. And it's going to be what? Right? Okay? All right. And then I can read this and figure out which amino acid goes in which order. And I only have one memorized. So the first one, AUG, is methionine. The rest I can look up in a book. But let's just say this one's proline, this one's leucine, this one's arginine, okay? And so on. Okay, you with me so far? We're building a protein. So this is our DNA, this is our messenger RNA, and this is our protein. Okay, now watch. What if I did this? What does that do to my protein? Totally. So instead, this is going to be an A. And let's say instead of arginine, this is now isoleucine. What this does is, because I've put just by one single base being wrong, it now gives me a different amino acid. And because I have one different amino acid that is not supposed to be where it's at, what it does is it now causes the protein to fold into a completely different shape. Now, a really good example of this is sickle-cell anemia. So usually what happens with sickle-cell anemia is you have one mutation that causes one codon to change, which causes one amino acid to change shape. And now, instead of having that red blood cell, that's your biconcave disc, you have a red blood cell that looks like a half moon with points on it. Imagine if I have more than just a point mutation. Imagine if I have multiple segments of DNA that are now mutated. What's that going to do to my protein? Probably destroy it. It's probably going to be no good whatsoever. And this is what mutations are all about. They can either cause proteins to change shape, or they can cause it that proteins can't even be produced, which certain proteins, now this is interesting, certain proteins in the cell can actually cause reactions in our cell to not happen. So some reactions just want to go and go and go and go and go and go, and these proteins step in and say, no, you need to stop. So these proteins, if they're not built appropriately, they let reactions just go too much and go too far, and when that happens, we call that cancer. Because we don't have proteins that will stop particular reactions from occurring. Any questions so far?